Hi everyone, Dr. Isom here, back to tell you about research methods that have been used to study the anatomy and physiology of personality. I'll be detailing the methods that personality neuroscientists, those are neuroscientists who study the neuroanatomy, structure, and function, as well as the neurochemistry, use to study personality. The oldest method there is brain damage, looking at naturally occurring brain damage, so accident victims, but also lesioning of animals. Phineas Gage is an example of a human who sustained, he's probably one of the most famous brain damaged humans, by the way, who sustained an injury, and specifically his injury was in the frontal cortex. And researchers were able to surmise from what happened to him before, his behavior before, compared with his behavior after, is that the part of the brain that he damaged, the frontal cortex, is crucially important in personality. So unfortunately, he's the poster boy for head trauma and how it causes personality change. But he did teach us quite a bit about how the brain is related to personality. So he's probably the most famous lesioned human, but there have also been studies that have involved lesioning of animals. There's a researcher by the name of Jaak Panksepp. I'll talk about him in a later part of the lecture. But he established that there are definite parts in the brain that are affiliated and associated with emotions. And in humans, those are the parts of the brain that are associated with some personality traits. Seizure disorder patients or epileptic patients have also been very influential in helping us understand that there are different parts of the brain that are active for different functions. In fact, there was a surgeon by the name of Penfield, and he operated on people who had seizure disorder when medication was not enough to control the seizures. The seizures in epilepsy are caused by scar tissue that is a result of things like brain infections, head injuries, tumors, or even strokes. As you know, the brain communicates by sending electrical signals in the form of action potentials. Those are sometimes referred to as neurons firing. When the brain becomes scarred, then sometimes it continues to fire, and that firing can turn into a seizure when it becomes uncontrollable. His surgical technique was considered groundbreaking because he created a way to go in and stimulate parts of the brain to understand what their function was, so that when he was cutting away scar tissue, he would not cut away important parts of the brain that were involved in necessary functions for the patient. He would go in with a tiny electrode with a tiny little amount of electricity, and he would stimulate different parts of the patient's brain. And the patients were actually awake because the brain doesn't have pain receptors. So all you have to do when you're doing brain surgery is do a local anesthesia on the scalp. There are no pain receptors in the brain, so Penfield was able to go in and stimulate individual sections or groups of neurons, and patients would be able to tell them what they would perceive. So he identified that there were certain parts of the brain that were involved in singing and certain parts of the brain that were involved in remembering commercials, for example. There are some pretty interesting anecdotal stories about Penfield surgeries. Anyway, his surgery provided an opportunity for us to identify certain parts of the brain that are very influential in different kinds of behavior. More recently, we've been able to access this new technology that allows us to see a window into the undamaged brain, specifically technology that allows us to take pictures of the brain in people that are awake, that are not sick. So we can identify whether there are differences in the size of different brain structures or the functioning and activity of different brain structures with behavior. We've been able to associate, for example, enlarged parts of the amygdala with more emotional behavior. And that has been made possible because we now have brain imaging techniques. So again, we can image brain structure, and some of the ways that we do that are with CTs, MRIs, and DTIs, which I will talk about in detail in a moment. But we've also been able to image the brain when it's working, when it's active. And the imaging techniques we've been able to use for that are EEG, MEG, functional MRI, and PET. So I will go into each of those in detail in just a moment. The first one is imaging brain structure. And two of the main ways that personality psychologists have related brain structures to behavior is by using computerized tomography That's this one on the left here, and also MRI. Now you'll see that the computerized tomography is actually pretty good. You can make out some of the different brain structures here, the gyri and the sulci. But the MRI is much better. It gives a much better resolution and allows a little bit more precision when relating the sizes of different brain structures to personality. So MRI gives the best spatial resolution. And by spatial resolution, I mean it gives the best picture, the most detailed picture of the aspects of the brain. This is an example of a CT machine. Perhaps you've had one. I know that they have these at local hospitals as well as the MRI machines. The patient would lay on this little conveyor belt and then get slid into the CT scanner, which is essentially an X-ray machine, and it takes many, many X-rays that are combined to form a 3D image of the brain. This 3D image can be used to compare size and location of different structures in the brain. The MRI looks very much the same. MRI stands for magnetic resonance imaging. You can see that there's a little conveyor and the patient lays on it, and then they are slid into this MRI scanner. And what this is, it's a very, very intense magnet. Here's an actual picture of one on the right. And what that magnet does is it sends magnetic pulses. In the case of a brain MRI, those magnetic pulses go into the brain and they cause atoms to spin in the brain. Not all of the atoms spin, only a percentage of them do, but they spin at a rate that's consistent with their density. And so different atoms in different parts of the brain spin faster than other atoms in different parts of the brain because they have different densities. And what that allows the MRI to do is to compile all of the different speeds of all of the different atoms in a three-dimensional space to provide a precise 3D image. Another way to image structure of the brain is with diffusion tensor imaging, or DTI. And this is a form of MRI. It uses the MRI machine, but what it's actually doing is looking at the structure in terms of the connectivity of neurons. So each one of these colorful little bands here represents groupings of axons in the brain. These are axons in the brain that connect neurons from one part of the brain to other parts of the brain. So again, diffusion tensor imaging is a technique that actually looks at the connectivity of different parts of the brain, can tell you what structures are connected. It can give you an idea of the size and the shaping of structures. And the way it does this is it measures the spin of atoms in water that are moving through the axons. So what DTI can do that CT and MRI can't is give us an idea of what brain structures are connected and what those connections are like in terms of size and location. You can study structure of the brain and anatomy, size, connectivity of the brain, but you can also study the activity of the brain, and that's very different. So in this case, we're wanting to see whether some parts of the brain are more active for some people and whether those levels of activity are related to behavior. So imaging can provide information on what parts of the brain are working and when they are active in terms of activity. If we want to know when parts of the brain are active, okay, and that means we want to know the timing, okay, we want temporal resolution, then our best bets for imaging the activity are EEG, electroencephalography, and MEG, magnetoelectroencephalography, which is very similar to EEG. If we want to know what parts of the brain are active, we would want to use positron emission tomography, or PET scanning, or functional magnetic resonance imaging. Whereas EEG and MEG will give you the timing, okay, it'll tell you when parts of the brain are active. It's not so great at telling you exactly which parts of the brain are active. It's measuring electrical signal or a magnetic signal from the scalp. Whereas positron emission tomography and fMRI can tell you with much more precision what parts of the brain are active, but they really can't give you much precision in terms of knowing when parts of the brain are active. Okay, so we'll talk about the timing part, knowing when parts of the brain are active first. This is when you want to know when the brain is processing information. And this can give you, EEG can give you information into the millisecond. Same thing with magnetoencephalography. If you want to be able to have precision in terms of the timing of when the brain is active, we would refer to that as temporal resolution. Okay, these are the best ways to measure brain activity with temporal resolution. On the left here, we have a gentleman with an electrode cap hooked on. These are the EEG electrodes, and these are the lead wires that then get fed into a computer. And so it's really not that cumbersome. It's just this little electrode cap. On the right here is the magnetoencephalography machine, and you can see it's much more cumbersome. I don't think that there's anything really attached to the scalp, but your head is within this big structure here, and what it's picking up is the magnetic fields that occur when neurons fire. EEG. What we're picking up with EEG is the firing of groupings of neurons in the brain. So we're recording electrical signals that are measured from the scalp. If you put a little metal disc on the scalp, a little conductive gel, it's amazing. You can pick up someone's naturally occurring brain activity. And if you were to combine all the brain activity that can be measured at one person's scalp, you could actually power a small light bulb or toy train. When neurons fire, they produce electrical signal, and that is exactly what EEG is picking up. Here on the screen, you can see what each electrode is picking up. Each electrode is picking up electrical signal. So each one of these little lines represents a different electrode over here on this woman's scalp. And it measures the timing of mental events, the timing of when parts of the brain are firing down to the milliseconds. So it's pretty darn precise. And what we do then is we look at the signal of EEG, and we can break it down into the different frequencies that are apparent in EEG. So there are different frequencies. We don't really need to go through all of them, except I do want you to know about alpha here. But first, EEG is composed of many different frequencies, and they're measured in cycles per second. That's how many times the sine wave goes up and then down in 1 second. We measure them in hertz. The one that's been studied most often in personality is alpha, and that's this one right here. Alpha is defined as a signal in the frequency of 8 to 12 hertz. So 8 to 12 revolutions per second of brain activity. It's what you see when someone is not asleep, but they're calm and they're relaxed and they're not processing information. If you're processing information, if you're intently thinking about something, your brain's going to look more like this. This is high activity. We call it beta. You don't need to know that, but this is what the brain looks like when you're thinking and processing. This is what the brain looks like when you're relaxed here in alpha. Alpha is produced when the brain is not really actively processing. That's important because we'll talk about some research that used alpha as a measure of the activity of different parts of the brain. If there is alpha, then that part of the brain is not actively processing information. If alpha is suppressed, this is an example of alpha suppression here. We don't really see any alpha here because the brain is processing information. Then that means that part of the brain is more active. Okay, magnetoencephalography, MEG, is very, very similar to EEG because when groupings of neurons fire in the brain, they not only send an electrical signal, but they also create a magnetic field around the signal that they're sending. And so MEG, instead of picking up the electricity, it actually picks up those magnetic fields. So it's very similar in that it can identify when parts of the brain are firing, and it can identify them down to the milliseconds. But it just does it a little differently. It doesn't involve placing electrodes on the scalp. It just uses sensors to detect tiny magnetic fields that are created when groupings of neurons fire. More ways of imaging brain activity. EEG and MEG were more focused on identifying when the brain is active, whereas these next two ways of imaging the brain are focused on what specific parts of the brain are active. If you want to really identify what parts of the brain are active, then you're going to want to use something like positron emission tomography, that's PET scanning, or fMRI, functional magnetic resonance imaging. These will tell you which parts of the brain are active. So we would say they have good spatial resolution, but they're not so great at temporal resolution because they require the imaging process to average across a few seconds. So you lose that temporal specificity. You lose the ability to measure the timing of mental events in milliseconds. Here is an example of a PET scan. This is an indication of which parts of the brain are active during a given time. And on the bottom is an example of a functional MRI. This shows the response of the brain when the person is looking at faces. You can see different parts of the brain are active versus the parts of the brain when someone is just looking at houses. So different parts of the brain are active depending on what stimulus the person is viewing. Okay, so fMRI is really thought to map brain activity. It can identify the parts of the brain that are active. But it does that by averaging across a period of time. It could be up to 8 seconds, 4 to 8 seconds. It's looking at the spinning of atoms. Remember, I talked about that with regular MRI. But it's looking at the spinning of atoms of oxygen. So what it can do then is identify where the oxygen is being used in the brain. And because we know that neurons require oxygen and sugar to function, what fMRI will tell us is the parts of the brain that are using the most oxygen. We can infer then that those are the parts of the brain that are active. Say you present a stimulus to a person and you identify that the frontal lobe is more active because there's more oxygen in that part of the brain, we can assume that that stimulus, whatever it was, activated neurons in the frontal lobe. Okay, the other way that has high spatial resolution, allows us to know specifically what parts of the brain are firing, is positron emission tomography. Just as I was saying that neurons require oxygen and sugar, fMRI measures the oxygen that's being burned in different parts of the brain. PET relies on sugar. So what happens with PET scan is the individual is given an IV of glucose, which is sugar, and that glucose has been tagged with a little radioactive tracer called 2DG. When the brain uses that sugar, the positron emission tomography machine identifies where that sugar is because it can see the radioactivity in the brain. So what you can see then in these images, let's look at the bottom here. When the person is awake, all these hotter colors, the yellow, for example, and the red, indicate places in the brain where there is more sugar because it is identifying where the 2DG is. That's where the sugar is being used in the brain. So this is the awake brain compared to the asleep brain, which you can see that's much less active because cooler colors mean less activity or less sugars being burned. However, if you've ever taken biological psychs like 36 or 125, which is behavioral neuroscience, you know that this brain, which is actually in REM sleep, is very active, as your brain is when it's in REM sleep. It uses quite a bit of sugar and oxygen because it's processing what happened during the day. So basically what positron emission tomography does is it shows you the parts of the brain that are using more sugar, and we can infer that parts of the brain that are using more sugar, those neurons are firing more rapidly, they're more active because neurons need sugar and oxygen to be able to fire. The last research method that I'm going to talk about right now is brain stimulation and personality. So we can identify differences in the size and the shape and the activity of different structures. But also another way that researchers have investigated personality in the brain is by stimulating different parts of the brain. By stimulating different parts of the brain, then we can identify how behavior changes after or during that stimulation. So these researchers then stimulate areas of the brain and then observe the effect on behavior. There's many ways to do this. For humans, what we usually see is called transcranial magnetic stimulation. That's what's going on over here with this guy. Or we have implanted electrodes, and this is not something you would just do every day. These are patients who have seizure disorder or Parkinson's disease that have had these electrodes implanted as a treatment. For humans, usually it's seizure disorder patients. Remember I talked about Penfield surgeries. His stimulation allowed us to understand what parts of the brain were involved in different functions like singing and memory. But also implanted electrodes for people with Parkinson's. There's a case of a woman, they implanted an electrode deep into her brain in an area called the basal ganglia, and the settings were off just a little bit because when they turned it on, instead of it treating her Parkinson's disease by stopping her tremors, it actually caused her to have several emotional reactions to the point even of feeling suicidal for a few moments until they turned it off. I mentioned transcranial magnetic stimulation. That's what's going on over here in this picture. And over here, these are the implanted electrodes that you would find in animals or in patients with Parkinson's disease who've had deep brain stimulation as a treatment. Here's that woman that I was mentioning, and this is the time course of when they turned it on. Here's when she started. She had the electrode implanted to treat her Parkinson's disease. And you can see what happens as soon as they started activating it. It was actually not in the correct place, or it needed a little bit of adjustment because she instantly started to feel this sadness to the point here 4 minutes later where she just felt extreme depression until the physician turned it off, and then she was back to normal. So as a result, one of the things that researchers identified was that the basal ganglia is involved in emotional functioning. I mentioned transcranial magnetic stimulation, and that's what you'll see here on this picture. It's a device and it's painless. It looks like a figure eight or an infinity sign. You hold it over certain parts of the brain and it will send a magnetic field into parts of the brain that can either excite or inhibit those neurons for anywhere from a couple of seconds for up to 90 minutes, which is pretty amazing because you could essentially turn off the ability for someone to speak. I saw a demo of that at a neuroscience conference once. But if you think about it even further, if we identified specific parts of the brain involved in things like empathy, you could cease empathy for a time period. If you stimulated or inhibit parts of the brain that are involved in visual processing, you could make that person unable to process anything visually, so essentially blind. But it does have a lot of therapeutic applications. I know it's been used to treat depression. It's still in the experimental phase right now, but that is one of the tools that researchers have used, stimulating different parts of the brain to see what behavioral changes occur. Transcranial direct current stimulation is similar, but whereas transcranial magnetic works with magnetic fields, transcranial direct current stimulation works with a small direct current, very small, I think a 9-volt battery, and it just sends an electrical signal from one part of the brain to another. And it has been shown to have some really interesting results. I know that there's actually a company that's using this currently as a sleep device, a device to cure insomnia. And there's lots of other applications that they claim that it can help. But it is another way of stimulating the brain so that researchers will have a better idea of what parts of the brain are involved in different behaviors and traits. So overall, there's quite a few research methods for studying the brain, many more than we had even 30 years ago. But they come with their own difficulties. For example, the fMRI uses a specific type of analysis called BOLD. There are other types of analyses, but this one called BOLD, it looks at the amount of oxygen used within a certain period. There's another one called perfusion imaging that works a little differently. But the overall problem with these is that we can understand parts of the brain that show more oxygen. But what we really can't know is whether the increase in oxygen in that part of the brain is associated with activation, okay, where the brain is actively using it right at that moment, or perhaps it's associated with inhibition, maybe recovery of those neurons at that moment. We can't really be sure what exactly is happening in those parts of the brain during the fMRI. We can identify that certain parts of the brain are more active than other parts, but we can't really say with any certainty that we know that that's definitely being activated or excited, because it may be, that part of the brain may be actually being inhibited. So it's really not as simple as it's made out to be. And in fact, there's been lots of criticism over the multitude of functional MRI studies that have come out since that technology was developed. And we also know that you can't just say that only one small part of the brain lights up or shows activity when presented with a certain stimulus, because we know that all parts of the brain are always active to some degree. And so that 10% cliche, you only use 10% of your brain at any given time, that is not true. And if you remember what I mentioned at the beginning of this lecture, that you have 85 to 100 billion neurons in your brain, and each one can be connected up to 10,000 times. So it'd be really difficult to argue that those parts of the brain that are shown to light up in most of those functional MRI studies, it'd be difficult to argue that those are the only parts of the brain that are involved in whatever kind of processing that person was doing. Another thing is that, let's say you show that there's a certain part of the frontal lobe that lights up or shows activity when you present individuals with some sort of an affectively arousing, some emotionally arousing stimulus. Just because that part of the brain lights up when they see that emotionally arousing stimulus doesn't mean that that part of the brain is only related to emotional arousal. It could be that that part of the brain lights up or shows activity for lots of things, and emotional arousal is just one of those things. So just because a part of the brain responds to a stimulus doesn't mean that that same activity is not necessarily associated with some other psychological process. Other things like maybe fear or excitement, other psychological processes could also activate that same area in the frontal lobe. And most researchers tend to only look at very small areas because, well, for one thing, this kind of imaging research is really expensive, and because some of the analysis requires them to limit their examination or their investigation to only small areas of the brain. One of the things that's important to remember is the neural context effect, and that's that there's no one part of the brain that is responsible for any one psychological process or function, because activity in one part of the brain is reliant on activity in many, many parts of the brain because of the connectivity that I was just mentioning. And certain parts of the brain are dependent on many other areas. Remember when I showed you the slide of the emotional structures in the brain? The amygdala and the hippocampus are incredibly connected. And so anytime the amygdala lights up, say in response to some sort of a threatening stimulus, we know that there's probably also activity in the hippocampus. So you need to consider that any part of the brain that appears to be active in a study is probably not the only part of the brain that is active in response to whatever that stimulus is. The last difficulty with research methods like this, imaging, is that they can be really difficult to use. I remember in grad school, it would take a couple of years to have the training to be able to do this kind of research. And then the equipment is very expensive and the maintenance of the equipment is very expensive. Okay, that's where I'm stopping with research methods, and we'll pick up with brain structures related to personality in the next lecture.